Hi, my name is Sharon Chen, and I'm a pediatric infectious disease physician at Stanford University. In this video, I'm going to tell you about antimicrobial targets to prevent microbial replication. This video is part of the Introduction to Microbiology series. In order to spread, microbes need to replicate. Scientists and doctors have taken advantage of this and identified means of stopping the replication with drugs. So, microbial replication is intimately linked with therapy. The learning objectives are to identify the targets of the major antibacterial, antiviral, antifungal, and antiparasitic medications and how they affect the microbes, to explain the concept of selective toxicity, and to explain common mechanisms of resistance to antimicrobials. Now imagine that these are two bacteria in your bloodstream. If unchecked, this is what will happen. Exponential growth, and it'll kill you or your patient. From the biology perspective, a famous microbiologist named Stanley Falco once said, "Every bacterium's wish is to be bacteria." Now I'm going to show you a movie of bacteria trying to grow in the presence of penicillin. You can see now when the bacteria replicate, they are lysing and replication stops. So this is what happens to bacteria when you infuse penicillin into your patient. An important concept in antimicrobial therapy is selective toxicity. What this means is that the drug has to be more toxic to the microbe than to the person. Many of the targets for antimicrobials are structures specific to the microbe and don't exist in a person, allowing selective toxicity against the microbe and little side effects to the person. In the next few slides, I'm going to discuss the major targets to fight bacterial, viral, fungi, and parasitic replication. This slide shows you the four main antibacterial targets: the outer cell membrane, the cell wall, the nucleic acids, and the ribosomes for protein synthesis. There are over 150 antibacterial drugs, or antibiotics, but if you can understand the targets, you can organize almost all the antibiotics into just a handful of groups. So in the slide, the blue boxes are the common antibiotics that you will use or encounter in the clinics. The orange boxes tell you about the target for the antibiotic. When we delve into specific microbes in the course, we will give you more details about these antibiotics. Right now, I just want to give you an overview. Through the course, you will learn about the details on this slide, and you can use this slide and the other antimicrobial target maps as a resource. On this slide, each of these four main groups of targets have been chosen based on the idea of selective toxicity. The targets are specific to the bacteria. So for example, bacterial ribosomes are different than eukaryotic ribosomes and make a good target. The cell wall structure is especially unique to bacteria and makes a very good target. Humans don't have this structure at all. The outer cell membrane is another structure not found in humans. It's also only found in gram-negative bacteria. So in this case, knowing the target of the antibiotic can help you remember what kind of bacteria the antibiotic can kill. Let's illustrate the concept of antibiotic targets with an example: penicillin. You have all probably heard of this antibiotic. Penicillin is in the group of cell wall agents. These are antibiotics that interfere with the peptidoglycan, which is the main structure of the cell wall. Here's a diagram of the peptidoglycan structure. Peptidoglycan is made of these parallel sugar chains. They're outlined in yellow, and they're cross-linked by short peptides. Penicillin blocks the enzymes that cross-link these short peptides. The enzymes are called transpeptidases. They are also known as penicillin binding proteins because penicillin binds to them. Now we're going to talk about antifungals. Unlike bacteria, fungi are eukaryotic cells and have many of the same organelles as our cells, so looking for targets is a little bit more complicated. However, fungi have two big differences from our cells. Like bacteria, they have a cell wall, but it's not made of peptidoglycan. The fungal membrane is also unique, with a specific lipid called ergosterol, instead of cholesterol, which is in our membranes. So the fungal cell wall and membrane are good targets, and many of the antifungal medications that you will use or encounter affect these two targets. There are other targets like nucleic acids and cytoskeleton, as you can see in the slide. A specific antifungal you might use or encounter is called amphotericin B, which targets the fungal cell membrane. It specifically binds to ergosterol and to itself to form a pore in the fungal membrane. Ions and essential molecules now escape through this pore, which kills the fungi. In this image, we are showing you the structure of amphotericin in green and ergosterol in red, and as you can see, it forms a ring with a pore in the middle. Now, unfortunately, the selective toxicity is not completely selective. Amphotericin can also bind cholesterol on our membranes, although the binding is not as efficient. When it binds to cholesterol, it forms pores in our membrane. As amphotericin is excreted in the kidney, this can lead to kidney damage. Renal toxicity is unfortunately a major side effect of this antifungal drug. Now, let's discuss antivirals. Overall, the development of antiviral medication lags behind development of antibiotics. This is because the virus is intimately linked with the host. Remember, it uses host cell machinery. Thus, similar to fungi, there are not as many targets that have selective toxicity. The slide here shows you the main targets for antiviral medications. Remember, there are multiple steps in the viral life cycle, and many of these steps are targets. I have grouped the antivirals to show you that they can target some of the main steps: entry and exit, replication of the viral genome, and processing of the viral proteins. You can also see in the slide that many of the antivirals are for HIV treatment. But the life cycle steps are very similar for all the viruses, and a target can be used to treat different viruses, not just one. So for example, protease inhibitors, these are the ones that inhibit processing of viral proteins, were first developed for HIV treatment, but now there are new ones that treat hepatitis C. To illustrate the point about antiviral targets, I'm going to use a medication that treats influenza. It's called oseltamivir, or you might have heard the name Tamiflu. This antiviral targets how the virus exits. It's a competitive inhibitor of neuraminidase, and here it's shown as the red protein on the envelope of influenza. In this close-up, you can see the virus is attached to the host cell via sialic acid. To release from the host cell after budding, neuraminidase cleaves sialic acid residues from the host cell surface. The virus is now released. Oseltamivir interferes with this release. Next, I'm going to talk about antiprotozoa targets. Finding targets for protozoa is also difficult. They are eukaryotes like fungi and ourselves, and they don't have cell walls. Some protozoa do share characteristics of bacteria and fungi, and this explains why some antibiotics and antifungals can be used. So for example, Giardia, Trichomonas, and Entamoeba, which you can see in the upper right corner, these can all be treated with an antibiotic called metronidazole. And metronidazole causes DNA damage when activated in anaerobic conditions. Now, all three of these protozoa lack mitochondria. Our cells have mitochondria, and we use aerobic conditions, which then never activates metronidazole. For some protozoa, targets have been found that are specific to their life cycle within one of our cells. This is the case for many of the medications to treat malaria. An example of drugs that target malaria when they invade our red blood cells is quinine, chloroquine, and mefloquine. When Plasmodia invade red blood cells, they live inside a vacuole, and their main food is hemoglobin. Digestion of hemoglobin leaves heme. This is a waste product, and it's actually toxic to Plasmodia. Plasmodia have then evolved a way of detoxifying heme by crystallizing it. Quinine, chloroquine, and mefloquine inhibit this crystallization. This results in an accumulation of heme that's toxic to Plasmodia by disrupting their membranes. Our final map is on antihelminthic targets, which are drugs that treat helminths or worm infections. Worms are divided into roundworms, the nematodes, those on the right, and flatworms, which are cestodes or trematodes, those on the left. As you can see in the slide, all the flatworms can actually be treated by one drug called praziquantel. It causes spastic paralysis of the worm. The antihelminths that treat nematodes are also neurotoxic. For example, parental causes spastic paralysis, and ivermectin causes flaccid paralysis. Other interesting targets exist. For example, some nematodes are actually killed by antibiotics like doxycycline and rifampicin. This is because they have an obligate bacteria in their guts, and they need this bacteria to survive. These nematodes are killed by killing the microbiota in their gut. A very important drug to treat nematodes is albendazole and its relatives. This drug interferes with the cytoskeleton. The movie I'm showing you shows microtubule cytoskeleton in a group of cells. One of the cells is dividing, and microtubules are very important in this division. Albendazole binds a subunit of the microtubule called beta-tubulin. This prevents polymerization and then kills the worm. Albendazole specifically binds to the beta-tubulin for nematodes, demonstrating selective toxicity. Our tubulin is different, and albendazole doesn't bind it. In fact, drugs that bind to our beta-tubulin are actually anti-cancer drugs. I have now showed you many of the drugs that we have to prevent microbial replication. Unfortunately for us, microbes have evolved ways to become resistant to these drugs in order to survive. This is the concept of antimicrobial resistance, and an issue that you will have to deal with many times in your patients. Resistance occurs with all antimicrobials. What I want to do is highlight 4 important resistance mechanisms. Each of these mechanisms can be an issue for many antibiotics. These resistance mechanisms can be transferred from one bacteria to another through horizontal gene transfer. In this image, we have a cartoon bacteria in the right corner, and on the left is an enlargement of the cell envelope of a gram-negative bacterium. The outer membrane is in red, the inner membrane in green, and the cell wall is in between in gray in the periplasmic space. The purple structures that you see are the proteins embedded in the cell envelope. So the first resistance mechanism that I want to tell you about is to not allow the antibiotic to enter. Gram-negative bacteria are already intrinsically resistant to many antibiotics because they have this impermeable outer membrane. Gram-negative bacteria have a channel, a porin, to move things through the membrane. Antibiotics against them must enter through these porins. Gram-negative bacteria can become resistant to an antibiotic by mutation of their porin, which then doesn't allow the antibiotic to enter. Another resistance mechanism is to push the antibiotics out once it has entered the bacteria. Bacteria have channels that go through the outer and inner membrane, and these are used as pumps. Bacteria can become resistant to antibiotics by alteration of these channels or efflux pumps. The result is that the antibiotics are pumped out and then can't kill the bacteria. Antibiotic resistance from efflux pumps are very important to know about because they can pump out multiple families of antibiotics, not just one. It leaves you with very few choices of antibiotics to effectively treat your patient. I unfortunately have to deal with these situations in the hospital quite often. Fortunately, there are some ways of minimizing antibiotic resistance. You will hear from me and other infectious disease doctors stressing the importance of using antibiotics wisely. A third mechanism of resistance is to degrade the antibiotics and render them useless. Specific enzymes can degrade the antibiotic. For example, beta-lactamases break the beta-lactam ring of many cell wall antibiotics. And here you can see beta-lactamases degrading an antibiotic in the periplasmic space where the cell wall is being made. A fourth mechanism is to alter the target of the antibiotic rather than affect the antibiotic itself. Mutations in bacterial ribosomes will still allow protein synthesis to occur, but not allow binding of certain antibiotics. Without binding, the antibiotic has no effect on the bacteria.